Let's take a final look at how we might regard a disease. What we call a disease and how it is caused depends quite a bit on evolutionary history and on the rate of environmental change. Now we're going to take a more detailed look at epidemiological transitions and diseases that are caused by mismatches between biology and culture. This diagram, which is from Graham Rook, shows the ancient history of a number of diseases. In the left-hand column, we have periods of history from the Paleolithic through the Neolithic and the Bronze Age to the Iron Age and the pre-industrial up to the modern. In the middle column, we have lifestyle. In the Paleolithic, it was small hunter-gatherer groups. In the Neolithic, it was larger groups. This is when domestication and the agricultural revolution occurred, and people moved into cities and they acquired pets, and they were living on farms with animals and with feces and mud and with untreated water. And in the modern situation, many people are living in cities with concrete, with soap, detergent, washed food. There's much less oral-fecal transmission. We have chlorinated water, less contact with animals. We have antibiotics and deworming. The kinds of microorganisms that were associated with us in these different parts of history were really quite different. Things that were present in early humans and had evolved with them for a long time and to which our immune systems were adjusted and whose removal creates an abnormal situation for the immune system include helminths, worm parasites, mycobacteria, tuberculosis, Hep A, gut microbiota, Helicobacter pylori, which interestingly causes stomach ulcers and stomach cancer, but also seems to protect us in some ways against other diseases. Salmonella, Toxoplasma, and Lactobacilli. Those are all things that we had, reliably had in our system in the Paleolithic. When we moved into cities in the agricultural revolution and we had a higher population density and there was easier person-to-person transmission, at that point our worm infections probably increased and there was probably more oral-fecal transmission, so there's probably more diarrhea. There were some new infections that probably came into humans at that point. So they've only been with us for about 8,000 or 10,000 years. A whole series of viruses, caliciviruses, rotaviruses, coronaviruses, paramyxoviruses, orthomyxoviruses, which include influenza, measles, mumps, smallpox, cholera, plague, and typhus. Those are all things that have probably only been with us for about 8,000 to 10,000 years. Then when we get into modern environments, there are big changes. There are fewer worms, there's less toxoplasma, less Helicobacter and Salmonella and tuberculosis, less hepatitis A, fewer pseudocommensals, and our gut biota is disturbed and it's less varied. So we have recently gone through two major epidemiological transitions marked here in red. One was about 10,000 years ago and one started probably about 2,000 years ago. What has been the recent history of disease? Well, in the very recent past, between 1950 and 2000, infectious diseases have declined. That was because of vaccination, antibiotics, and hygiene, and autoimmune diseases have greatly increased. This is the incidence of infectious disease you can see in the United States. You can see rheumatic fever, hepatitis A, mumps, and measles all going down. And at the same time, multiple sclerosis, Crohn's disease, type 1 diabetes, and asthma are all going up. If we look in space, we can see there's a negative spatial correlation of pathogens and type 1 diabetes, which is an autoimmune disease. So the red places on the planet are places where there are quite a few worms and there is leprosy and there are fairly high incidence of large eukaryotic parasites in people. The yellow places are places where there is very little of that and there is very much less type 1 diabetes. So that's one spatial correlation. If we then look across countries And so here we have the incidence of tuberculosis and the incidence of type 1 diabetes. Here's Finland and Sweden and Norway and the UK over here. Very little TB, lots of type 1 diabetes. And over here we have places like Poland and so forth that have not very much type 1 diabetes, but quite a bit more tuberculosis. One of the common things that clinicians notice if they go to serve in the tropics is that they don't see much autoimmune disease when they go to the tropics. So that's a suggestive pattern. We saw one in time, this is one in space. There is some experimental evidence on this. So these are cases in which a particular kind of genetically modified mouse, it's called the non-obese diabetic mouse, it's a model of diabetes, has been raised in two different conditions. In the left-hand panel, we see the development over 80 weeks of age of the incidence of diabetes in two generations of these mice. You can see the two generations are right on top of each other. And this is in a conventional breeding situation where they do have a microbiome. They have bacteria in their system. And the incidence rises to about 50%, and it takes about 35, 40 weeks to get there, say 35 weeks. If these mice are then raised in the next two generations, generations 3 and 4, in a pathogen-free environment, two things happen. One is that they much more rapidly acquire diabetes, so already by week 20 or 25, most of them have it, and 75 or 80% of them get it, rather than 50%. So you can see that pulling all of the microorganisms out of the gut of this mouse model of diabetes greatly increases the rate of diabetes. We also know that worms will prevent autoimmunity in animal models. Here is a table showing infectious agents that will prevent type 1 diabetes in that kind of mouse that we've just looked at. Schistosoma, Helicosimoides, Trichinella, Mycobacteria, a bunch of bacteria, Mycobacterium and Salmonella, and even a virus, mouse hepatitis virus. These are all affecting T helper cells in some way or another in the immune system. If we look a little bit more broadly, where a worm infection or the product of a worm prevents autoimmunity, we can see that for Schistosoma, Trichinella, Trichuris suis, which we will hear more about, Helicosimoides, and a product of an acanthocephalin, they can actually prevent or reduce Graves' thyroiditis, encephalomyelitis, experimental colitis, experimental bowel disease, and collagen-induced arthritis. So those are all cases where having a worm in the system seems to be modulating and downregulating autoimmune disease. Well, that raises the issue, might we want to have worms in our system? Is it unnatural not to have them there? Well, here are some worms. This is Schistosoma, which causes schistosomiasis. Here's Trichinella, which causes a very nasty disease. That's what you get from eating uncooked pork. Here are a couple of worms that are fairly often used in experiments with rats and mice. And the one of these, which really seems to have the best combination of characteristics for use in therapy, is the pig whipworm, Trichuris suis. It has little or no pathogenic potential. It doesn't multiply in our bodies. It can't directly spread to another human. It has a limited colonization. It's asymptomatic, and so forth. On the other hand, it does stimulate our immune system. So people have been trying autoimmune therapy with the eggs of these pig whipworms. One of the objects of therapy is Crohn's disease. Crohn's disease is an inflammatory bowel disease. It has persistent diarrhea, abdominal cramps, pain, fever, and fatigue, and death can actually result from Crohn's disease. So the first study was simply a very preliminary one. They just had 4 patients that had Crohn's disease and 3 with ulcerative colitis. All of them improved after ingesting 25 embryonated pig whipworm eggs. So that was just kind of a first step, just to make sure that giving them the whipworm eggs wouldn't damage them. Then a bigger study, 23 of 29 patients with Crohn's disease, got 2,500 eggs every 3 weeks for 24 weeks, and they had a significant reduction in their symptoms. Then, better, there were 54 patients that got a double-blind, placebo-controlled trial. They had active ulcerative colitis from Crohn's disease. 43% of them were treated with worm eggs improved, and we compare that to the placebos where only 17% improved. So that looked pretty good for Crohn's disease. What about multiple sclerosis, MS? Multiple sclerosis is an autoimmune disease that is really a horrible disease. Your own immune system is attacking the nerve sheath on the axons in your brain. It does so in many different ways. Some people get a very mild form of MS and can live with it throughout their life. Perhaps their arm goes numb for a while, but then they get their control back over their arm and they do well for decades. I have a friend who has it. Other people get MS and it's degenerative and it progresses rapidly and it leads to loss of control over walking, talking, breathing, bodily functions, and death comes relatively quickly and is pretty horrible. So treatment for MS is something to be highly sought for. Most of the current treatments are treating symptoms and they can slow down the process, but they're not getting at the cause, which is a dysregulation or an abnormal response of our own immune system. Correale's work in Buenos Aires has a very interesting set of results that may point to a possible solution. What you see here is from his first paper in 2007. On the x-axis, we have 60 months. On the y-axis, we have the number of new and enlarging lesions in the brain of patients, all of whom had multiple sclerosis when they entered the study. They were, however, divided into two groups. Some of them were infected with worms and some of them were not. And what Correale followed was how rapidly did the symptoms of multiple sclerosis progress. Well, the ones who had worms, the uninfected ones, hardly progressed at all. They had very few of the symptoms developing. The ones that did not have worms got worse in a hurry and more or less stayed there. So they had on average 1.5 lesions in their brain, and they got there fairly quickly, in less than a year. Interestingly, after following these patients for several more years, after 5 years, one of the patients developed a really bad response to the worm infection. So it was one of the patients in this group that was doing quite well in terms of MS, but had a worm infection. They treated that particular patient right here to rid him of his worms, and look at what happened to the multiple sclerosis symptoms. So they pull the worms out of the system, and very rapidly, within a period of 1 year, that patient has as many lesions as the group that is not infected. So treating the parasites accelerates multiple sclerosis. Now, what kinds of mechanisms are involved in these sorts of interactions? This isn't just for MS. This is for all of these sorts of atopies and autoimmune diseases. Well, the immune system consists of different populations of cells. Interestingly, they all come from the same stem cell in the bone marrow. They then have different developmental trajectories. They develop into dendritic cells, or into macrophages, or into B cells, or into different kinds of T cells, or into natural killer cells. And they communicate with each other. That's what these dotted lines and these symbols mean. So the interleukins are molecules that are sending signals from one cell to another to help regulate cell populations and to induce particular functions in the immune system. So if we look at what worms seem to be doing, they seem to be blocking the communication between B cells and eosinophils, and it's the eosinophils whose degranulation produces allergies. So worms seem to be blocking allergies. They are also modulating with strong induction, that's these dotted lines, or with weak induction, that would be these thicker lines over here, communication between T helper cells and T regulatory cells of various kinds, as well as between dendritic cells and T helper cells. So the effect of that disruption of communication by the worm is basically to affect the incidence of diabetes here with this particular interleukin and the incidence of experimental colitis and arthritis. In other words, there's a complex signaling network between immune cells in the body that is being specifically targeted by worms. Now, when we think about that, it could work through simple homeostasis. The infection could just simply be distracting the immune system. It could be competing for cytokines, for growth factors, or just for space in the lymph tissue. This is plausible, hasn't been ruled out, but it hasn't been demonstrated. There might be downregulation of the immune response to avoid inflammatory damage of sustained worm infections in such a broad way that allergic and autoimmune reactions are dampened. This is possible, but there's good evidence for more specific interference with signaling. There could be an elevation of overall circulating antibody levels, both specific and nonspecific. That would then dilute allergen-specific antibodies, and it would saturate receptors on mast cells. This is possible, hasn't been ruled out. But what is probably much more relevant is specific interference with signaling, as we have seen in this diagram here. Now, why is it that worms evolved to become really master manipulators of vertebrate immune systems? They send their eggs out of our bodies into a very hostile environment with very low chances of colonizing a host. They have to send thousands to millions of eggs out to have a reasonable chance of getting one surviving offspring that successfully infects the next host. That must be done over a long period of time. They therefore must persist and also thrive and grow large enough to make lots of eggs in hosts whose immune systems are doing everything they possibly can to get rid of them. So worms, which look like they might be a good thing for autoimmune disease, are in fact a double-edged sword. They protect against autoimmune disease in the developed countries, but in the tropics, they produce worm infections. And because of those infections in the tropics, they increase the susceptibility of their host to TB, AIDS, malaria, and other infectious diseases. And they reduce the protection that's offered by vaccines. It's actually harder to get a good response to a vaccine in a human that's got a worm infection. You can go to this issue of parasite immunology if you would like to read further on those problems. Now, we referred to the fact that the immune response can be problematic. This is what the inflammatory response looks like. If the vertebrate host, humans, had remained as sensitive to worm infection as they would have been the first time in evolutionary history that they were ever infected by a worm, then they would produce a strong inflammatory response and try to get rid of it. That inflammatory response would look something like this. The pathogen would be triggering macrophages to release signaling molecules, cytokines and chemokines. Those are then picked up by the cells in the walls of capillaries. There would be vasodilation. In other words, the cells actually loosen up and they let the fluid that's in the bloodstream and the cells that are in the bloodstream out into the tissues. That causes redness, heat, and swelling in the tissues. And the cells that are in the immune system that are specialized on finding and attacking pathogens migrate out into the tissue, and they release chemicals that can cause pain. We're all familiar with this. We get a scratch, it turns red, it hurts, right? This is what's going on. So this response is disrupting local physiology. And if it doesn't actually succeed in getting rid of the pathogen, then it's imposing a lot of cost at no gain. Well, worms know how to avoid this problem, right? So they cause chronic infections, and therefore, if we had an inflammatory response to worms, it would have a high price. This can be seen in the 1918 flu epidemic. About 60 million people died worldwide in the flu epidemic. The mortality rates were high among young adults that had healthy immune systems. They were the ones who could mount the really strong response. They produced a strong inflammatory response to the flu virus. Their lungs then were intensely inflamed by a cytokine storm and filled up with fluid, and that then enabled a secondary bacterial infection to come in and kill them with pneumonia. So that's just an extreme example, but it brings home the point that chronic diseases of aging and other things that are the result of inflammatory mechanisms are being caused by a reaction of the body, and continual inflammation is going to cause real problems. For example, atheromas, the fatty plaques that we have in our arteries, those are caused by damage to the wall of the arteries that produces an inflammatory response. There's local tissue modification and the plaque starts building on that substrate. There are senile plaques in the brains of Alzheimer's patients that seem to be growing around areas of inflammation. The risk of heart disease is correlated with the concentration of inflammatory molecules. As I've mentioned, the infection by Helicobacter pylori causes both gastric ulcers and raises risk of stomach cancer, and it also increases the risk of a heart attack by 20%. Another piece of evidence indicating the importance of inflammation in these processes is that anti-inflammatory drugs reduce the risk of several diseases. So if you were taking things like aspirins or statins or NSAIDs, then you are preventing heart attacks and GI tract cancers. And you not only are lowering blood lipids with statins, they also have an anti-inflammatory action, helps to avoid the risk of arterial plaque and heart disease and the risk of Alzheimer's. So all of this is basically to say that it's quite plausible that we have co-evolved with worms that cause chronic infections so as not to overreact to their presence, cause inflammation in the tissues surrounding the worms, and then pay a high price in consequence, because we wouldn't be able to get rid of them because of the fact that they've been so good at evolving to manipulate our immune system. So to summarize, our bodies have co-evolved with quite a diverse community of bacteria and parasites. If these are pulled out for a variety of reasons, by hygiene, by vaccination, by clean water supplies, our immune system responds inappropriately, and it produces atopies and autoimmune diseases. The mechanisms that mediate these responses are now partially understood, and worm therapy offers some hope.